For the talk, I will need help from a friend and a colleague who contacted me and said, you know, I have some amazing data from animal and cell studies. Let me see if I can go back. So that's Hilde Nielsen, Professor Hilde Nielsen. So she will be with me and also tell something. And you have your mic, so she can tell a little bit more of what she found in both before we started the study, but also within the study. So we are a small group, small country, but we are following all with individuals with AT annually or biannually. And the two studies I want to present for you are both the study from the Netherlands. They started ahead of us as we planned to do the OR study, and both studies are published. So I want to both present these two studies and also compare them. In DNA, ATM, let me see, AT is a DNA repair disorders, and the DNA damage response signaling is active in AT. And there has been a long story of scientific work that has provided evidence that also NAD is reduced in Ataxia-Telangiectasia. Do you want to add something here? And from Hilde's work, we see that there's a chronic activation of PARP, and which leads to this depletion of the NAD plus in the ATM knockout cells. And restoring cellular NAD levels would restore, with nicotinamide riboside, will extend life and attenuate the neuromotor function loss in animal models of AT. And this is Hilde's work. And she came to me and said, we should do something now. Hilde, you have been working with Willem Bohr and other groups in DNA repair and aging. And can you help me to explain this one?
Yeah. Can you hear me? No?
Yes.
So with Will, we modeled. We used animal models, different types of animal models, to see, first of all, basic scientists, we are really, really concerned with mechanisms, not just what happens, but why does it happen? And the motivation for that is that we try to sort of break these pathways that leads to something bad. In this case, mitochondrial dysfunction and neurodegeneration. So we did that using our mouse and C. elegans, with a little roundworm models. And that we could map a pathway. And then as the next question, we asked, so can we break this circuit in any way? So we used small molecules, for example, olaparib, which is used for treating breast cancer with a BRCA-defective breast cancer, to break this step. And then we also used as a 2 inactivator to break that, or boost that step, which is sort of a rate limiting factor for, it's really important to generate new mitochondria, get rid of bad mitochondria. And in addition, we find that NAD plus levels are overconsumed in this situation, because this is a great PARP NAD consumer. If you cannot break or repair the break, the backup function is the PARP, and it goes on cycles and try to protect NAD break. And that leads to an extortion of the NAD pools in the cells. So this is the central integrator. So the easiest thing is, if we can add more NAD, does it help? And yes, we found that it helps. And this is really showing just if you add nicotinamide riboside, the NAD levels improve. If you add SIR2 inactivators, improve, and if you add olaparib, it improves. So we could also show that that rectifies the neurodegeneration and really improves the health of the animal models. And based on that, parents picked it up. We got a lot of anecdotal reports from families in Israel, particularly in the US, and also elsewhere in the world. They contacted Wilbur and they contacted us, and we got descriptions of their experiences using NAD. And that was what really gave us the funding to do the study, because we went to the health minister saying that, okay, we need to do this in a study. We need to really know whether it works or not, and then that's where it starts.
So these two studies from the Netherlands and us are both single-center, open-label, observational studies. So we have no control persons. They both had requirement was that the individual should have a genetic diagnosis of AT and more than 2 years or more than 3 years in our study. And the exclusions were liver disease and cancer treatment or cancer, active cancer. And we had the endpoint, which was change in whole blood NAD levels, and the second endpoint was changes in biochemical markers and neuromotor function. And I'll say some few words about the neuromotor function testing, because it's different tests out there, and your kids and family members may have gone through all these tests when they go to the pediatric neurologist or the neurologist. Actually, what we have made is it's a combination of all these tests in our study to make the testing result more complete. And usually the physiotherapist play with the child, so it doesn't, really realize that it's tested. And we try to do this as quick as possible, and we have the video recording to go back to form the scoring. So the Netherlands study, it was a 4-month study, and then 2 months without supplement, and then they did the testing of these two neuromotor scales, SARA and ICARS. And they had altogether 24 patients with AT enrolled, and 22 were tested with these neuromotor scales. And in our study, we have a 2-year study. We did some safety in the beginning, started with a low dose and increased the doses, and had a recording of the potential side effects of the supplement. And we had initially 14 enrolled, and 14 was invited, 13 was included, and we had we have 3 withdrawals, and I can say something about that later. And later on, we have continued, so we still do the study in the youngest patient, and also one old patient, and we can present some data from that as well. And what Hilde has shown is that the NAD levels is increasing with the treatment, with the supplement. And we also can, when we compare these two studies, we have no change in, no severe side effects. That's the most important. And there's no change in total blood count, white cell differentiation, liver enzymes, renal function, AFP, and a marker called a neurofilament light chain. And we didn't find it either, none of the changes. And in the study from the Netherlands, they found a change in immunoglobulins. We didn't see that in our long-term study. But we had one who had to stop because of loose stools. One that really had a better function of the bowel, was so successful and wanted to continue with the supplement because of that, and one had some discomfort in the stomach. So what I told you is that we had 14 invited. 13 enrolled, 2 were later excluded because of cancer and infections, and then 1 with this loose stool problem. And we had additional 4 included in the continued study. Can you say something short about this?
Yeah. This is just showing that we do see that NAD levels increase, and also the other conversion products of NAD, the products that you see when NAD is consumed in the body. So that is good, that's what we expected. But what we feared is that you also get other things happening in the blood that we didn't expect. And the good thing is that we really didn't see that. Over the course of the study, we only saw the increase in the metabolites, or the small molecules in the blood, that we expected.
Can you say something about what you did here? I was told you about the neurofilament light, that we had great expectation to that, but we had no real change in that during the supplementation period. And how about
yeah, so it's a lot about talk about interferon, type 1 interferon signature in AT, and we measured this by looking at the RNA in the blood. And we do see that the pink and the green here shows that there is an inflammation or a type 1 interferon signature going on. And if you have 6 months of AT, that what you see in the AT is kind of ameliorated a little bit. It's a little bit milder.
Red field is normal. The green is before supplementation, and the blue is with supplementation. So it's not a big difference, but it's some difference here. So I'll go back to the results of the 2 studies. So in the study from the Netherlands, they found that there was a difference between those who were at baseline and those who were treated, and even they could even see that when they stopped the supplementation, some of the positive findings went back. And they didn't show any changes related to age, AT phenotype, and gender, but they found these serum immunoglobulin level changes. And in our study, we found great changes in some domains, some neuromotor domains, but not in all. And when we compared what we found to historical data, we have followed individuals with AT for many years with these neuromotor test scales, then we found that here a decrease in scores is a better function. And here you can see that in SARA total and SARA coordination, it's easy to see that it's an improvement. And when we put these data we have from this study plus the ones we are following now, together with the historical data, it's easy to see that Niagen improves neuromotor test scores. So the green dots and lines are the treated or the individuals with supplementation, and the gray dots and lines are the historical controls. So what we, both in 18S total and SARA total, there is a big difference, and it's also interesting that it is in the elderly individuals, not only in the youngest one. But it's also interesting that there's no improvement in the walking, but it's in coordination and eye motoric function. So what we can conclude is that NR supplementation is easy, safe, non-invasive intervention, improves coordination and eye movement in AT. It's observed across ages, all genotypes, but we only have classical ATs in our cohort, and there's no significant improvement in gait, stance, and sitting, and no changes in immunoglobulin levels. So we would conclude that we have to do more studies, broader studies in more groups, and also to do controlled studies to really conclude here. But on the way, parents are giving their children this supplementation, and it's good with these 2 publications to show that it seems to function. So then I want to thank Hilde, who contacted me, and the group, the clinical AT team, and also the medical student who did a lot of the work, and also all the colleagues at Hilde's lab and her collaborators who have done the the laboratory, both the proof of concept, but also tested the extended patient samples. And in the end, thanks to all the families who contributed here, because it's a huge effort to perform a clinical study. Thank you.